Naresh so far?
Okay, we are live now. Anyone here? Good evening, Sarojini. Good evening, Mallikarjun. So we have two people who have joined. Nagaraj, can you see if the people have joined? I mean, sufficient people have joined. Nagaraj?
Yeah. Only
Can you
two
check?
people have joined.
Okay, fine. We'll wait for a couple of minutes.
Yeah, sure. Now 7 have joined, so you can start it.
Okay.
Yeah.
Good evening, everyone. My name is Mohammed Yunus. The voice is echoing. Nagaraj.
Yeah. Just a second. Okay.
Yeah, my name is Mohammed Yunus, and I am co-founder of the PERF Foundation. This is the foundation that me and Nagaraj have started recently, and I'll be your moderator for today's webinar. This webinar is a part of our monthly worldwide webinar effort with an objective to bridge the industry and academia. This is supposed to happen regularly on monthly basis with speaker from different area speaking about their expertise and skills. The idea is to share the knowledge. Our speaker today is Dachana Murthy. Dr. Murthy received his master's from BHU and obtained his PhD from University of Madras, Chennai. He has more than 2 decades of experience in cell and molecular biology and worked at various national and international research laboratories. He has published several national and international publications with high impact journals and has been speaker at several conferences and has a patent also on his name. He contributed significantly in establishing a microarray lab at Institute of Genomics and Integrative Biology, New Delhi, and worked extensively on oligo, cDNA arrays, and Affymetrix gene chips. As a guest scientist, he contributed to the identification of a mitotic checkpoint kinase involved in p53 independent apoptosis at Professor Hank's lab at Germany. Further, he also worked on a project, DNA vaccines, that is gene therapy, as molecular medicine at Dr. Yang's lab, Taiwan. In India, he worked at DRF, that is Dabur Research Foundation, toward development of targeted therapeutics for the treatment of cancer, for which he holds the patent. He also worked at Orchid Chemicals and Pharmaceuticals Limited, Chennai, where he was part of a drug discovery team focusing on small molecule inhibitors for cancer therapy, and contributed significantly in the development of an orally active isoform of HDAC inhibitor for the cancer therapy. At present, he serves as a consultant biologist at iNext Biosciences for artificial intelligence, machine learning, and deep analytics-based modeling of therapeutic antibodies, enzymes, and small molecules. I would like to welcome Dr. Murthy and wish all the participants will have a fruitful webinar. Dr. Murthy, please, you can take the session now.
Yeah, thank you, Nagraj and Eunice, and thank you, Nagraj, for my introduction. Am I audible? My voice is clear, Nagraj?
Yes.
I can hear you.
Okay, now I will start sharing my screen.
Sure.
So share my audio. Share. Okay, this is my first slide. Is it visible to everyone? And my audio is clear, Eunice? Okay.
Yes, we can see you.
Okay. Yeah. And first of all, I would like to thank Dr. Nagraj and Eunice for arranging this webinar as an industry and academic collaboration. The first one by Patil sir from academics, and the second one I am from representing the industry thing. So my organization is INEST, which is AI-powered biosciences, which is involved in developing AI technologies for drug discovery and antibody engineering. So before I start my presentation, I would like to thank particularly all the healthcare workers and researchers who are working around the globe for their effort in controlling the pandemic and such that we are nearing towards the end of this pandemic, and hopefully the new year, that is 2021, will be free. We will be free from this pandemic, and we will be back to our work in our labs. And going to the presentation outline, I divided this presentation into two parts. The first one is on, just a second. So the first one is on artificial intelligence and machine learning and deep learning and data science. So basically, I will be explaining the basics of artificial intelligence. What does it mean? And what are machine learning, deep learning? What is the basic difference between these terminologies? And what are the different methods and algorithms which are used in this artificial intelligence, machine learning, and deep learning techniques. And then I will also touch upon big data. So now the big data science is exploring very fastly. And then on data mining, which is very important for us. And the next part will be on drug discovery, how this AI, ML, and DL is used for the development of newer drugs. And also, I will touch upon the pharma challenges at present we face because of artificial intelligence, and some slides in which I will show on the pharma industry and as well as the AI company collaboration, which at present going in a big way to develop novel therapies, especially for COVID-19. And then I will, just a second, conclude my presentation and giving a perspective to the pharmacologists who are viewing this, what kind of knowledge we can have or develop in order to become a data scientist, and if you want to explore your career in data science. So to start with this traditional drug discovery and development, generally it has two phases. The first one is drug discovery, and the second one is drug development. The drug discovery is nothing but the identification of target, and which lead to the discovery of new drug against that particular target. And once we identify the target and validate the target and validate the lead, then we will take that molecule into preclinical phases, which involves a lot of in vitro and in vivo studies. In vitro, we do cell-free and biochemical assays, whereas in vivo, we do animals, mostly animals and rodents. And where we will study the ADMET properties, that is absorption, distribution, metabolism, excretion, and toxicological profile of the drug, of the lead molecule. So once we do all these things, we assess that whether this particular molecule has the potential to be escalated for clinical use, clinical trials. Then we go on to submit investigational new drug application, and then followed by clinical development, which consists of three phases. Phase one, which is a small toxicity and safety and kinetics study, and phase two, which is against the large cohort of patients, around 300 patient population. We have testing the drug efficacy, and then we go for phase three, in which we take a large cohort, very large, around in thousands, and then compare with the existing drug and the placebo and confirm that the particular drug is performing well, and then it can be submitted for as a new drug application, following which approval it will come into the market. And once it also come into the market, we have a pharmacovigilance, in which we study the long lasting side effects and drug interactions and so on. We keep on generating the data. So this is, again, I put preclinical discovery in a shorter thing, because this is the area in which the AI and artificial intelligence is much of use. The first one is identification and validation and developing an assay. Now we have bioinformatics and genomic and proteomic tools for identification of lead discovery, and the lead is then taken up by the medicinal chemist for a synthesis, which could be either structure-based or ligand-based drug design. And the medicinal chemist then chooses from the hit, he chooses a particular lead molecule. And then we check for the in vitro drug activity and cellular models and drug mechanism of action in pharmacological model. And also we do a lot of in vivo studies wherein the ADMET properties is examined. And once if a particular lead molecule crosses all the hurdles in the particularly in these studies, then it can particularly escalate to the next level for clinical trials in human. But as we all know, the drug discovery and development is not a easy task. So far, the at present the trend is that it needs a lot of resource, lot of time, lot of money, and lot of human effort to discover and develop a new drug. And apart from that, even though such a long term time, around 10 to 15 years at present, it takes a new drug to come to the market. Most of the drug fail in particularly in clinical trial, particularly in phase 2 clinical trial or preclinical testing or to the validation, which causes lots of money loss, a lot of monetary losses to the inventor. And also this leads to the high cost of the drug in the market, and also the long cycle time and the limited success in the clinical trial. So basically, for the past 150 years, we have been working with this rationalized drug development system, wherein we understand the human pathology and the disease pathology very well. And also, we have been developing a lot of drugs which are already in the market. So we have a lot of data with us, either from the discovery or from the preclinical testing or from the clinical trial. We have a lot of data generated either by the pharma companies or to a small extent by the academic labs. So we have a lot of success and failure stories with us. Now the question is, can we able to use this data of success and failures, and we can able to predict a drug like compound such that we can reduce the time and the effort and money in developing a new drug. So this is the point at which we have the newer technique which is coming, which is artificial intelligence. This artificial intelligence is nothing new to us, because it is now widely used in all walks of life, like voice recognition and self-driving cars and many more. And whenever you go to a Google search, it is based on a machine language learning algorithms, and all of the world which we live today, the internet is flooded with artificial intelligence and its technologies. So now the question is, what is artificial intelligence? Artificial intelligence, in a simple word, artificial intelligence is nothing but making the machines to think like humans. So we can also call it as human intelligence can be brought into the machine, such that the machine will have an intelligence to think, like to go for which shop or which product the customer needs, or which paper you are looking in the Google search. It itself make a decision and it gives the result to you. And next comes the machine learning. And the machine learning is again a subset of artificial intelligence. The machine learning is nothing but it puts lot of statistics and data analysis tools with the help of algorithms, and these algorithms which in turn learn from the data which we supply, and then it gives, when we give a new data to it, it will say that this particular data can be classified as a drug or not. So that is what the machine learning is doing. Machine learning is the application of statistics and data analytical tools to analyze the data. Here, the programming is not done to find a solution. That I will come to a later point. What is programming and all. And next is the deep learning. Deep learning is again a subset of machine learning, which is an advancement of machine learning, which uses lot of networks. And in simple words, it makes the computers to think like how the human thinks. Like we have a brain, we have a neural system, we have a thinking capacity. But if you impart the same character, that is a learning character, how the brain learns, the same way the machine can also be made to learn and make decisions. That is called as deep learning. Here also we have modeling algorithms. So now I will go to the basic timeline of AI. This AI, artificial intelligence, it's not a new technology as most of the people think of. It started very long back, around 80, 90 years before. The first one was Alan Turing. He was a British soldier, a very exceptional computational biologist. He came up with the Turing test, and then he asked the question, the first one to question that whether we can able to ask the machine to think and like intelligent people think. And later on, Arthur Samuel invented, and he also the coined the term machine learning in 1952 itself. And after which there are many progress that has been made. Particularly, the theoretical progress has been made. The theory and the statistics and other things were made. However, the applications were very limited, like ELISA and the Blue computer, IBM Watson. Particularly in the 1990s, we have the IBM Watson playing a chess with Gary Kasparov and winning the game. So once that happened, then only in 1990s only the machine, the artificial intelligence came to the public, that people think that even the computers can think and play like human individuals. And then now at present, after 1990 to 2000 to 2011 and all, there are lot many, like after the Google came, Apple and Microsoft, all of them have developed lot of machine learning, use the machine learning technologies in their mobile computing as well as in search engine algorithm. Like in mobile computing, now we have Cortana from Microsoft, Siri from Apple. All these follow the machine learning algorithm. So okay. So now I'm going to tell the very basic difference between artificial intelligence, machine learning, deep learning, and DLS, data science. Hereafter, I will be using AI, ML, and DL such that it will be easy for me to introduce it very fastly to the listeners. The first one is AI, which we can see that, for example, it thinks like humans. Like we have a deep IBM Deep Blue, which won chess against Gary Kasparov, and electronic games and the mobile video games which are available in the internet. All of them are artificial intelligence. They could able to make guess that what move the opponent is going to make, and then play accordingly. Whereas machine learning, when we think, it is mainly algorithm based, in which it learns categorically and then predicts, like Google search engine, which we use, and Amazon recommendations, which we have, and email separating the spam from the normal mail and all. And then we have a deep learning. This is again a subset of machine learning, wherein we have AlphaGo. This is again by Google. These are using artificial neural networks, which are very capable of taking very complex tasks and make predictions. Like if in AlphaGo is from Google, if you give a sequence, it will predict the 3D structure of a protein sequence if you give the primary sequence. Similarly, the speech recognitions. software, face recognition software, self-driving vehicles. These are very complex and are being done using deep learning techniques. And next comes the data science. Data science is subset of all of them, because data science is nothing but it is a science which combines computational as well as mathematical tools together. Like we have mathematical means, we have probability, statistics, linear algebra, all of them are taken into account. And the data science are the data scientists, or those people who can able to design different algorithm, which could be either deep learning or neural network or machine. They will write codes for developing a newer algorithm, which is used for machine learning or deep learning. And as I told in my previous slide, even the artificial intelligence was long back, it was came into picture, but only in the 20th century, that is the present century, which is there is a much more talk about artificial intelligence, because now we have a big data. The big data means we have a lot of data points with it because of the advent of computers from 1980s to 2000. We have computerized everything, and we have lot of data points for each and every activities which we do. For example, the drug discovery, we have a database of drugs. For example, zinc database, which has around 50 million, 300 million compounds are there. In PubChem, we have several compounds are there. Similarly, we have so many PK, PD data for which has been generated and has been computerized. So there is an explosion of data is there, and we need some techniques to analyze the data. And second one is the speed and power of computation has drastically increased. For example, previously we are talking about central processing unit for computers. Now we talk about graphic processing unit, which is the core of deep learning techniques, wherein the predictions, wherein the analysis is mainly done as a graphic processing unit, whereas the prediction is done by the CPU or the central processing unit. Because nowadays we have Intel iCore i9 generation, that is 10th generation uh, chips are available for even for personal computing. And then we have the cost of computing become very low because of the cloud infrastructure and distributed computing. Distributed computing means you need not to have a very expensive servers or expensive uh, um, machines, computers, like with the extended random access memory or extended computational capabilities in your own lab. But rather, there are many cloud companies are providing this computing uh, space in which you can able to do your analysis over the internet, and you can have the data with us. So all these three things made this artificial intelligence possible now, and this is an explosion of data, or now it is the time for data or data science. Now we come into the learning. Machine learning, as I told here, the computers are not being programmed, because previously we heard about computer programs, like different programs we have used for our, for example, Gmail. If you take, it is a program for handling your email. It will do only, it will open the email and it will give you the input of how many mails you have received and all. They are programmed to do a specific task. But when compared to that, the machine, on the other hand, the machine learning, using the machine learning, the computers can able to learn from the data which we supply. For example, we have data, 10 or a data of 100 compounds, and some of them are potentially inhibiting a particular drug target, and some of them are not. And if you give this data to the computer, then the machine learning algorithm will learn from it. So what is responsible for the compound to be inactive, what is responsible for the compound to be active, it can learn from it. And next time, when you give a test data, when you give to the trained model, then it can able to predict that the given compound, whether it is active or inactive. So we have input, we have training data. It will learn from the input data and select features and can tune parameter to generate a model, and this model will then predict the test data whether it is a drug or not. So before going into it, so a short introduction for algorithm, because we are not computer scientists, but just to give a very basic definition of algorithm is nothing but it is a set of instruction in a particular sequence given to the computer to solve a problem. This is mainly logical sequence. Logical sequence means yes or no, or a kind of logical steps are given to a computer uh, with the help of a computer programming language. So here again, this programming language is the one which the computer can understand. So we have to write the code in a particular programming language. The common programming language for machine learning are R, Python, Java, JavaScript. So many other, but these are the three more common languages for programming languages for writing codes for machine language. And also these languages also have modules, which we call it as machine learning libraries. These are different modules which are available. Lot of machine learning libraries are available. These type of libraries are nothing but a particular library will have a particular algorithm and will do a particular task, and they support a variety of machine learning algorithm. And now I will discuss about what is machine learning and what are the different types of machine learning. So the first one, we have a classical machine learning, which is again divided into unsupervised, supervised, and then dimension reduction. So this is the classical learning algorithm. And then we have a reinforcement learning algorithm and a model. And then we have neural network and deep learning methods. In this, again, we have neural networks like CNN, RNN, GAN, and autoencoders, and so on. So I will be going briefly about what each and different methods, and also discuss different algorithms which comes under different method. The first one is a classical machine learning. So it consists of methods and algorithms. So the methods is supervised learning, unsupervised learning, or semi or reinforced supervised learning. So here, as I told, the machine learning will learn from the data which we are supplying. Once it learns, then with the help by using a particular algorithm and model, then it can predict that whether the given data, what it is. So the first one is supervised learning, in which we give the data in the form of labeled data is given to the the model. Labeled data means, for example, if you take an Excel sheet, we have labels, like first label correspond to the gene name, second label correspond to the gene expression levels, third label corresponds to the disease in which the genes are associated. So if you give a labeled data to the model, then it is called as supervised learning. And again, in supervised learning, there are two type of classifications there. Two type of models, methods are there. One is regression and another one is classification. A regression is nothing but for a scalar value. For example, if you have you heard about fitting a trend line using regression. So if you have a linear regression, for example, if the data points lie in a straight line, then we can able to extrapolate that particular line, and we can able to find the y-axis or x-axis for a particular data point. That is the regression. And we have a classification. In this case of classification, here based on the structure, it will able to classify. So I will discuss these two things in detail in the coming slides. And then comes the unsupervised learning. In case of unsupervised learning, the data is given as such. There is no labeling is done. There will not be any label. But in case of unsupervised learning, the method will learn certain pattern. It will look for certain patterns which are present in the data, and then it can able to predict that and understand the data by looking at certain pattern. And once when we give a new data to it, then it will classify that either this could be this yeah, drug or not a drug, something like that. Here again we have a clustering and dimensionality reduction. On the other hand, reinforcement learning, here it is also, some people call it a semi-supervised learning. Here in turn, it is like learning from reward. For example, here we have a data, we feed into the model, and the model predicts something, and then a reward is given to it. If it currently predicts it, then a reward is given to the prediction. Then from that, it then the predicted one again, it again goes and it goes in a cycle, and then it keep on predicting with the reward, and then it comes up with the prediction model. And here I have mentioned different algorithms. For example, decision tree, random forests, artificial neural networks, deep learning. Even deep learning will come under this. Bayesian support vector machines, nearest neighbors. These are all are examples of supervised learning algorithm, supervised learning method based on classification. And in unsupervised, we have a clustering, we have hierarchical clustering, k-means, and also in case of dimensionality reduction, we have principal component analysis, SVD, and ICA. So first, I will briefly discuss about supervised and unsupervised learning. Supervised learning, in which the data are labeled, and we have the algorithm used to solve is based on classification or regression problems, whereas in unsupervised the data are not labeled. It will look for some highlight or some pattern. It will look for some hidden pattern from the vast amount of data which is given to it, and then they will try to either cluster that particular data. Cluster means classify that particular data, that this particular data sets or may behave like a drug. This particular set of data, this particular class of compounds will not behave like a data drug. So in supervised, again, I told again we have regression and classification. In regression, as we all know, this is for we supply a uh, pre-labeled data to the model. So here, in case this classification is that based on the prediction is based on the pattern. For example, here we have a share market. For example, it is like going like a bull or going like a bear. So here we can able to, the prediction is based on a classification. So the classification with the algorithm will classify either this or that. Whereas in case of regression, this is like the normal regression which do correlation regression, which doesn't have output or numbered outputs are there, and this will show that particular table which contains different data sets, like relationship between height and weight of the people. For example, if you have a data like data of 50 male individuals of different weights, and then we give, and each weight will correspond, we will have a label like they're obese. Particular 40-year age old male is obese and his weight is 80 kgs, and 30-year-old age male, his weight is 20 years. So it will make predictions based on the label we give, and then based upon the weight and age, it will predict that the given individual is obese or not. Next, in case of unsupervised learning, we have the input the model is input given it's a clustered thing, and the model will then separate based on the patterns which are present here. For example, here a football, basketball, or a tennis ball, all it separates. Here again we have similarly we have patterns, like we have a combination of patterns, and it classifies the clusters the pattern based on the structure. So this is an example which we all have come across, the unsupervised hierarchical clustering. So where we can see here there is a particular a red color which shows a particular expression level of a particular gene. This is a microarray data. So and the green color is low abundantly expressed, and the red color is highly expressed. So these are particular cell lines. You can see that different type of, for example, Hodgkin's lymphoma. These are different labels, like different cell lines, and you can see that when this particular data, cell lines and expression pattern is given to the model, this will predict that this particular cell line, that a Hodgkin lymphoma patient have these many genes which are overexpressed, that is in red color. And so similarly it forms a pattern. It looks for the pattern and it classifies it based on the pattern which is hidden in the data. Say for example, here it nicely classified as this is low expressing and this is high expressing genes in Hodgkin's lymphoma, in mantle cell lymphoma, or other type of lymphoma. So again, as I told, supervised, and these are the different algorithms for regression and also for clustering. We have different algorithms. And when compared to the drug discovery, for example, unsupervised is mainly used in classifying the disease subtype, or in case of finding in the target discovery, finding a suitable uh, pattern in a drug targets. And also supervised learning, in we have labeled data like ADMET properties, we have labeled data, and a diagnosis, for example, certain labels with the disease, it can be used for diagnosis. And reinforcement learning, where we have drug design like de novo drug design and other things are predicted by using these models. So this is again the reinforcement model. So where we are feeding this data, raw data, into the model. So this model, based on the selection of the algorithm, it selects a particular environment, selects a particular data, and it rewards that, and again it is fed into the algorithm, and then select. This cycle goes on until all of them are clearly separated as distinct patterns. So to sum up, supervised has known labels or output. Unsupervised is finding patterns and gaining insight from the data. Whereas reinforcement, we have a making decision based on the previous experience and reward. So these are the commonly used machine ML methods in pharmaceutical research. I will come a little about this artificial neural networks in the coming slide. So we have K nearest, support vector, mission decision tree, random forest. So these are the different time, and depending upon the data sets, we can use it. For a smaller data set, we can use support vector machine or decision tree. For larger one, we can use PCA or random forest. And these are the advantages and disadvantages of different ML methods. So generally in workflow for the machine learning is that this is a general workflow. First is the data collection, either labeled or unlabeled data. And then we need to curate the data, particularly we need to prepare the data to be able to given in a CSV file that will be appropriately categorized, such that the model will easily take and understand. And then we have to appropriately choose a model. Model means either supervised or unsupervised. And then also the algorithm we have to choose based upon the data set size and then training has to be given based on the model and algorithm to the model, and based on the training the evaluation is done. Once the evaluation is done, then find the parameters has to be tuned such that there won't be any overfitting or underfitting, or then we when we have gained a confidence, then we can go for prediction. The prediction is by giving a new data set to the machine models. It will predict that whether it follows as a thing what we ask or not. So here again, the choice of machine learning algorithm. So which machine learning method we need to choose for which data. For example, if the data is very simple and the clear features are there, we can go for classical learning. And if there is the data, for example, the environmental data, space data, and so many data which doesn't have any numerical input, that can be go for reinforcement learning. And real problem is ensemble methods, and very complicated large data, and so many unclear features. For example, if you have hundreds or 2000 drugs which different pharmacopores, this is very complicated data, and we cannot future find some features in the drug pharmacopore. So these things can be made fed into deep learning methods to find suitable answer, like whether this could be a druggable like molecule or so. Now we move to the artificial neural networks. Previously, we saw about machine learning. Now we go into artificial neural networks because this artificial neural network form the basis of deep learning. This artificial neural network, as the name has, it is the function of the artificial neural networks are similar to the function of a neuron which we have. So towards your left hand side, we have a neuron, we have an a body, and then axon and dendrites, and then the axon tail. So whenever the through the help of axons of the previous nerve, the signal is received, processed in the body, it is transmitted to the next neuron with the help of axons, and then through the dendrites. And this is how our neurons work. In the same way, we have an input. So here the inputs are x1, x2, and x3 data, and some weightage is given to each input, and then it is fed into the body or a node, we can call it as, and it is then processed. For example, certain calculations were made, such a weighed output is given. Weighed output is then passed on to the next neuron, and similar this process go on until we arrive at a solution. So these are called as artificial neural networks. And we in artificial neural networks we have three different types of layer. The first one is input layer, hidden layer, and output layer. The input layer is the one in which the data is fed into, which like a dendrites of the biological neuron. And next one is the hidden layer. So the each hidden layer consists, these are the different neurons you can think of, and these are also called as nodes. And the number of nodes in a hidden layer again is determined by a trial or error approach. So you have to do a number of how many nodes you want to have based on the trial and error. So the for predicting the number of nodes you have, then the data it predicts with the highest correlation and lowest error can be taken as the optimal for selecting a particular number of nodes for a hidden layer. And then final layer is output layer, which consists of output. So this is an example, this I will skip. And here again, in artificial network, neural networks, the advantage is that unlike classical machine learning algorithms, in which, for example, the linear methods, linear regressions, and all, which will learn from only from the linear data, whereas in case of artificial network, we can take a non-linear data. Data need not to be linear, and even the complex relationships which exist between the data points. So these type of data we commonly see in drug discovery, in pharmaceutical formulation. We have a very complex relationship. For example, the association of a particular genes, say for example diabetes, a particular gene is complexly associated with many factors like environment, sex, age, and also the food status, race, ethnic status. So many complex relationship is there. So in order to make a decision, we can rely on this artificial network, neural network data. So this has a high capability of learning, and also the unseen relationship in a particular data will also be learned by this kind of artificial network, and thus making this predictions more reliable and generalized. And also the distribution, for example, in probability distributions, we look for Gaussian distribution, but here the data need not to be distributed. Uh, but even if they are not properly distributed, they can also be analyzed by artificial neural network. So this is the main differences that, such as animal studies and other things, for disease pharmacology, all can be studied using artificial neural networks. Next come the deep learning. So the deep learning is again a machine learning technique. As I told, this is a subset of machine learning technique. The deep learning is nothing but it it thinks like a human brain. So in this case, based on the neural network, how does it thinks like a human is based on the neural network. It permits a machine to train itself and perform a particular task. So it can easily train itself and perform a task. The difference between artificial neural network and deep learning neural network is that number of nodes. Here the nodes are very simple, where if it has more nodes in deep learning neural networks. So this is the basic difference. For example, in traditional one, we have an input and we have to extract the futures. Futures means, for example, in a particular drug database, the future is molecular weight or the log p value, log d value. Different futures has to be extracted and given to the traditional machine learning algorithm in order to predict and give an output. Whereas in case of deep learning algorithm, if we can give an input and itself makes a future extraction by itself, and it can also classify the data itself and can give the output. So this elimination of future engineering is very useful when the user doesn't know which futures to extract, but he wants to analyze the data. So the examples of deep learning algorithms, a common one involves the artificial neural networks, particularly useful for the numbered data. Convoluted neural networks, this is particularly useful for the image data or video data. Images means microscopic images, the pathology slide, histopathology slides. All the images can be used to predict whether the particular slide section taken from the patient is having, it can be classified based on the pathology. We can able to classify whether he has a disease or not. And similarly, the video data. So this is common with the data which we commonly use for mobile computing. And then we have the recurrent neural networks. For example, if you have a time series data, for example, if you do dosage studies for different time points and different time limits and the different study points or their time points are there, then these kind of networks are very useful. And next one is generative adversarial network. This is particularly for discriminated data. For example, for discriminating a large set of data, this is widely used. So, particularly this type of GAN is particularly used in de novo drug discovery. For example, here it consists of two neural networks, generator and a discriminator. The generator, you supply a database of a particular drug. So then the discriminator will see that the take a particular drug A and then see whether it is following the category of A or not, and then it tried to discriminate with the other one B. And so whether it is a real drug or a fake drug, the discriminator then uh, classifies. Once if it is a real drug, then it goes through the discriminator. This goes as a cycle, and the discriminator can no longer distinguish the fake drug from the real ones. So that's how the model is built, and the new drugs are being identified. So this is the overview of machine learning algorithms. We have different types of data. We can have an individual or combination of algorithm can also be used. You can also combine two or more machine learning algorithm to have analysis, and complex nonlinear relationship can also be modeled, and it can also from the complex relationship it can also make accurate predictions. So next, I will briefly touch upon big data. So big data is nothing but the data which is high in volume, high velocity, variety of program, and veracity. That is, trueness of the data is very high. So all these four V forms a component of big data. So the big data, as the name implies, the data is very big. For example, we have a compounds. We have the chemical compound space is very huge. Out of which the compounds which we have synthesized so far will be around 10 raised power 8 compounds which has been synthesized so far. For example, those many compounds are there. So we have human genes, we have around 20,000 to 30,000 protein coding genes are there. Their functions are hundreds and thousands of functions, different types of cells and different types of disease. So the all of these are big data for us. And these data, even though they are very big, they are heterogeneous in nature. Heterogeneous means this chemical data is different from the gene sequence data. The gene sequence data is different from the gene function data, and this data is different from the cells and also the disease. So you can see that there is the data is very complex, so and very large amount of data is there, and analyzing such data using a classical computer programming programs, or using excel sheet or ms access database. So this will not help us to classify these compounds based on certain user queries. So in this case we need to go to artificial intelligence or machine learning approaches to listen to the data, interact with the data, and extract knowledge from the data. This is, for example, the omics data. That is, omics means genomics from genes, and transcriptomics from the mRNA, and proteins from proteomics, glycomics, metabolites from metabolomics, which comes from mass spectrometry, and the genomic data come from sequencing. Particularly nowadays we have next generation sequencing, and again transcriptomics, mRNA expression studies, we have DNA microarrays, multiplex pcrs, and next generation sequencing. Similarly, proteomics, we have protein profile to mass spectrum, 2D mass spectrum, 2D gel electrophoresis, and then mass spectrophotometric analysis, and post-translation modification like phosphoproteomics and glycoproteomics. We have a lot of data, and also we have lipidomics. So we have variety of data generated by omics technology, and in addition to that, apart from this omics data, we have network, because we individually know how the gene is expressed, how the RNA, particular RNA is expressed, or how the particular protein plays a role in a particular disease, how it is a metabolite is metabolite, a drug is metabolite. So, but these are not happening separately, but together as a network. If we make a network of complex things, like for example, network from cell, tissue, organs, and human system, and from the chromosome and the phenotype, if you make a complex network, then it becomes systems biology, in which the analysis still becomes more complex. The data is also big, and the data type is also very complex. So this is about the complex data force, and that is one type of data. Then in pharma research, what are the other big data which we have? For example, these are the drug target, the drug metabolic network data, which I already discussed in the previous slide. Apart from that, we have data from patents, publications, online chemical database, chemical libraries. We have a lot of PubMed has millions of documents, and we have metabolite information, drug molecules. So much of data is there. So how we are going to handle this data? And in case of drug discovery, what are the different type of data we have is that for target identity and validation, we have druggability, chemical biomarkers, and in case of hit and hit to lead optimization, we have library design, visual screening, drug design, and in case of preclinical, we have ADME and tox data, and in clinic, we have different clinical patient segregation, resistance to therapy, and so many data which we have from the drug discovery itself. So, so this is the preview of the drug big data in drug discovery. Next is the data mining. The data mining is nothing but, so given such potentially huge data, how to mine the data, how to get the information that we want from such huge database. For example, if you want to data, you simply go to PubMed and type 2 diabetes. It will give some hundreds and thousands of articles, but you don't know what to see, which article to read, what are the genes which are involved in it. So we don't have any idea about how the data is present in different le- uh, the papers which are coming under a PubMed search. So the huge data is there. So the data mining techniques is nothing but using the machine learning, statistics, and artificial intelligence to analyze, and the database technology to mine the data to get the useful data. For example, so this is IBM WhatsApp concept recognition. So what it does is that this particular IBM Watson modeling algorithm, it go and searches all the patents, for example, all the chemical document. It searches all the chemical images present in the document. And then, for example, if you type in what are the different, for example, if you type volume, which is nothing but diazepam, then it searches all the documents and give what are the different names under which the diazepam is, the trade name is diazepam is reported, under which the trade name it is sold. So like that, we can able to it can able to see the read from the chemical structures which are present in the literature and can give you useful information. Similarly, for example, this is an example of multiple sclerosis. Similarly, the Watson, for example, for the Medline, for multiple sclerosis, there are around 26 million Medline abstracts are there. Then the query is, for example, if I want to know what are the different type of genes which are associated with multiple sclerosis. Then if one needs to use Watson, then this is a network graph based for the multiple sclerosis. It goes and reads each of the document and picks up the each of the gene and put it as a network, and which is easy to read. This is an example of data mining. So now I come to the next part. So far we have discussed about different artificial intelligence, machine learning, other thing. This is the last part, is how this is useful for drug discovery and development process. So in this case, the colored one or the one which wherein we have lot of data, in which we have this artificial intelligence has a transformative impact. So particularly these are the omics and system biology. We have lot of data, and we have target identification. We have lot of target validation is phenotype screening data. We have a lot of data, and in preclinical research we have lead identification. Lead identification means either structure or ligand based. We have lot of chemical compounds, lot of data associated with it. And for clinical and preclinical, we have a limited set of data. The data which we cannot call it as big data, but data is there. And then we have repurposing. For example, at present we are thinking of repurposing the drug for the use which is not mentioned in the label. So for which also we have a lot of data. So these are the different areas in drug discovery process wherein artificial intelligence can play a major role. So that traditionally we used to work with this for we have a compound synthesized medicinal chemist. We do a high throughput screening, and then we directly again go to ADMET studies in animals and look for histopathology and so forth. And meanwhile, we do structure activity relationship because of the advent of QSAR models. Then because we have now the structures of the target also available in the PDB, so we take it as and we dock it and we find score it, and based on the score we again synthesize, fine tune it, again we put for assays. And once this is come, then it is taken for clinical outcome. This is how the drug is discovered at present. This is a traditional approach. But when we go to the artificial intelligence, and there are many areas in which artificial intelligence can impact. For example, the for predicting the drug target and validating the target. For example, for validating a target, we have a lot of target crystal structures are available. We can do in silico docking and other things and predict the druggability of a particular target and signaling and so on. Similarly, in case of ADMET property and SAR property, we have a lot of data wherein AI can help in uh, finding a suitable drug-like candidates. And then in clinical trials, we have selection of patient population, stratification of patient population, and drug repurposing, and also for pharmacovigilance. So these are the different areas in our drug discovery wherein the artificial intelligence can be efficiently put to use such that we can reduce the time, effort, and money in developing a new drug. So I am quickly going to give you some examples of how, because this is a very broad topic. At the end, I will give you some references which you can go through to exactly read about what are the different algorithms used and what are the different predictions have been made. But in general, for target identification and validation, so we have omics data and heterogeneous data from target, disease, and drug associated data sets we have. And for compounds, we have combinatorial library. Large number of compounds are being synthesized. Previously, we have high throughput screening. um, Based upon the combinatorial chemistry, we have a high throughput screening that is purely experimental. High throughput screening is purely experimental, where you synthesized thousands of compounds and you put them for preclinical testing, wherein we can avoid those HTS, high throughput screening, because of AI, wherein we can able to model certain train a model and then predict that particular drug is worth to be taken for preclinical study or not. And also the drug like properties also, we have so many like log P, log D, hydrogen bond, rotatable hydrogen bond, polar surface area, so many parameters we know for the existing drugs and other non-drugs. which we can take. We can use the AI models and then predict in beforehand before going into preclinical studies. And in case of preclinical, we have so many biomarkers which are being predicted. So these biomarkers can be taken, and the classification. For example, the cancer drug, we have a lot of drug response signatures are there from gene expression studies and other things. And clinical endpoint prediction of biomarkers. This is also particularly in preclinical market. We have to more the number of biomarker we have, more efficiently we can predict the usefulness of a particular compound in preclinical development. So we have biomarker dimension from single cell data and proteomic and other data. In clinical, we have cellular based in oncology, phenotyping in oncology, and precision measurements in the tumor microenvironment. We have pathology and slides and other samples and patient recruitment. So many areas in which we can employ artificial intelligence and have a data. And this is again a nice paper from Nature Reviews Drug Discovery, wherein they put different machine learning techniques like supervised and supervised, different algorithms, and using these algorithms, what are the different areas people have tried and reported. This is a 2019 paper, very good paper which appeared in Nature Drug Discovery Reviews Drug Discovery, where one can go and you can see how this particular model or algorithm is used to predict, for example, here ADME property or association or quantitative structure-activity relationship and so on. So in drug discovery, we have structure and ligand-based drug discovery, and we have a de novo drug design. This is a very new topic in which we don't know. We just start from the scratch. We design one by one pharmacophore and make it as a drug based upon the target. So if the target structure is available, we go for X-ray and NMR structures are available, then it is well and good. Otherwise, we go for homology model or based on different type of ligand. We have a molecular descriptors, pharmacophores, and QSI models, which we can use for predicting potential drug candidates. This is again in clinical trials, we can stratify the patient and increase the clinical outcome based on the mechanism of action of a particular type. Similarly, in preclinical, we have a molecular profiling. The very good understanding of a drug at the molecular level will help to develop a suitable biomarker and hence the mechanism of action, which will helpful in patient stratification and higher efficiency of clinical trial. And this is where the drug discovery in the healthcare come together into come up with a personalized medicine. We have genomic data, we have pharmacology data, we have patient data like electronic health record. All these things and individual data and other cohort and population studies, like these three main database are set into training, and then prediction can be made such that it will help in stratifying the data based on the individual or cohort or population, and we can validate and advance such that in the near future we can able to stratify the patient based on the genetic status or the molecular status, such that it will pave way for development of personalized medicine. At present, we have many targeted therapies for cancer in such way. So again, this paper from Nature Materials, this also one can go through how this all this target discovery, solubility, hug, ADME properties, skin permeability. So you can go and interestingly, you can read these papers and find out how they took the data, how they model the data, and how they got the predictions for the new moiety. So based on which you can know how these different techniques are being, your machine learning techniques are being harnessed for the drug discovery. These are the different computational based tools for drug discovery. the AlphaFold, which is from Google for a 3D structure protein 3D structure prediction, and similarly from prediction of novel properties, we have a neural graph print and delta vina deep talks for toxicity predictions. These are the publicly available database. And next, this is an example of publishing a drug target inhibitor for a particular drug target, DDR1. This is dioxin receptor, which is meant for treatment of cancer, a particular type of cancer treatment. The target is selected, and this kind of reinforcement learning, they can go for rapid synthesis, which molecule to synthesize. They predict with the help of target. They predicted this target, and even the in vitro assays they could do in 46 days, and then publish the paper in Nature Biotechnology. So traditionally, it takes around 2 to 3 years for hit to lead, but using this particular approach, they can able to less than 2 months. They can able to make predictions and uh, make a suitable drug-like molecule, which they reported in Nature Biotechnology. And similarly, this is IBM Watson. This is again a AI company from IBM. So they offer different type of. For example, I have put here Discovery Life Sciences. They can if you want to look for niche which target. If you have a target, whether you can if you take this target, will it succeed or not? There are so many questions we have when we want to work with the target. Similarly, when we want to how to comprehend that particular disease targets and how to take the compound further with different preclinical parameters. So at the constant context of this many big data, they can able to, but this is a paid software, and they can able to, they are saying that they can help in different aspects like creating target for you, composing data, pathway analysis, comprehend all these profiles, and create a data domain specific content they will create for you, and they can extract the data also from this big data infrastructure for you to go with. So these are the challenges. Some few slides are there. The first, why even though the artificial intelligence has largely grown, there are many challenges are there. First one is that the data governance. For example, these are so much of data is there from biochemical, analytical, sequencing data, for which there are different. Each drug has a different single experiment has a different protocol, and so much data is there. So far, the data are not properly governed. I mean to say that they are not being properly structured, and majority of the data are with the pharma industry, which they protect it. They are not available publicly, except some of the data which is available public. So this data governance, that is integration and management, is a potential pitfall at present in artificial intelligence, the application of artificial intelligence, and lack of single unifying problem. For example, if you want to discover a particular drug, there are so many factors are there: targets, disease, preclinical, clinical trials. So many inputs has to be made, for which we need to concentrate on different vendors, which decentralizes the resources. Whereas this is not in case of ordinary AI. For example, the common task like Netflix or Amazon, they have a single task. Like they want to give the content according to the nature of the audience. But here the problem is not single, but it is a very broad problem, wherein a lot of AI mediated challenges are there. Next is this insufficient skill set. So for machine learning, we need computer science and mathematics and maths and statistics. For data science, we have a combination of knowledge of business in case of business. In case of science, we have and computer science and maths and statistics. And in case of AI, the large part of science background is needed. Particularly in our pharma industry, we have a lot of PhDs who have a domain expertise, but doesn't have the AI like machine learning and data science exposure, So if we are to ready to accept and learn these uh, uh, techniques and it will be very useful to understand the data because we understand the drug more efficiently than any other field in science. And traditional scientific approach, because previously we worked on hypothesis, because we are very good in postulating hypothesis based on a certain hundreds of data in a particular data sets. But now we have millions of data where where we need to generate and assess data based on artificial intelligence. And absence of investment, many companies are very slow in developing or investing in AI because of a variety of problems like uh, so here again in case of the companies which the US, the majority of the companies are focused in US and UK and Asia and European Union. Less number of companies are investing in AI. The pharma companies are investing in AI. For example, the investors are more in EU, and the companies are also more in EU, sorry, US, when compared to Asia or Europe. So hereafter, we need to think of data-driven drug discovery and healthcare. This is again the list of FDA-approved biosensors which is linked to artificial intelligence. Not only the drug discovery, the artificial intelligence is used, but also the biosensors. Now we have the Apple Watch, which has inbuilt ECG measurement and blood glucose level, and so many biosensors which are inserted in the body are being monitored constantly by artificial intelligence with the help of Internet of Things, IoT. And because our drug discovery programs are very, drug discovery and development involves many different areas. We have different companies which are coming up from hit to lead, PKPD studies, trial optimization, antibody, biopharmaceutical, small molecules. There are a number of companies have come. They are helping developing AI technologies for these therapeutic area. And these are the leading pharma companies, AI companies, investors, and tech corporations. So you can see that mainly the Pfizer and Roche, Amgen, Merck, GSK, Novartis, AstraZeneca, Sanofi, all of them are now moving into AI-based drug discovery approach. So the 10 pharma companies, which I have mentioned here, they are partnering with the benevolent, particularly Johnson& Johnson or IBM Watson. Already Pfizer and Novartis, they have collaborated, and Berg, Exscientica, and Insilico Medicine, Synapse, and Path AI. So because nowadays the companies themselves comes and collaborate with this. And as I showed in the previous example, like IBM Watson comes up with the AI solution for different drug discovery problems. So in such case, if you have a large amount of domain expertise and also an understanding of machine learning, then you can able to utilize these functionalities. So in conclusion, so the machine learning approach and it increases the drug discovery development pipeline, and the impact is more on omics and imaging data, and the data are very becoming very big, and more powerful algorithms are required to generate improved output, and interesting applications are going to come in the near future. And now we have a healthcare setting combined with drug discovery, which will pave way for the personalized medicine. If you have artificial intelligence put into place to predict, and then can also be, as I told you, it comes from the sensors and wearables, which will help us in the future to understand disease in deep, and then develop or tailor treatment according to the disease or biochemical parameter. So, in short, it is accelerating the drug development process, reducing timeline, and reducing the high attrition rates which is associated with traditional drug discovery. Uh, so, as such, this drug discovery, artificial intelligence and machine learning is going to certainly change in the next 10 to 15 years. It will become very common as how the QSA or computer-aided drug design is common nowadays, which was developed 20 years before. And 20 years from now onwards, this is the age of artificial intelligence in drug discovery. And this is a perspective for the pharmacologists who are the listeners of the today's session. We have a good understanding of pharmaceutical sciences and drug discovery research. So this we have, but we need to develop a strong background in molecular and cellular biology because we are now, apart from phenotype-based drug discovery, we are now going to molecular-based drug discovery. We are going to do where we need to have a good understanding. We need to have a technical aspects on computers, and now the other technologies like genomics and the imaging technologies are there, like NGS, next generation sequencing, proteomics, microscopy. We need to gain knowledge. Gain knowledge means what are the data points they produce and how they will be analyzed. So these kind of data, this kind of reading we need to do. And knowledge of public domains, we have a lot of public domains like zinc database, chem embl, so many database, disease database, genomic, clinical trial database. So we need to have a common knowledge about these domains, and then such that we can given a problem we can look into this database, because we can't don't have access to pharma company database. We are now rely on this public domain database for doing machine learning, for developing machine learning. And one of the main important thing is that biostatistics, because all this data is analyzed based on certain statistical tools and probability. So we need to have, and programming languages to learn is the common programming language R and Python, and also the IDE, that is developing environment, is PyCharm and RStudio, which we can learn. We need to learn at least either of R. Python is very easy when compared to R. And big bioinformatics and big data analysis. So now we have, previously bioinformatics, when we are in 2000 and 2010, bioinformatics was at the peak. Now slowly it is now converted into big data analytics, where all are using AI and ML for big data. So hereafter we will be shifting to big data analysis. And apart from this, we need to have artificial intelligence, ML and DL learning from different platforms like Coursera, EDX. Many platforms are there online wherein you can take this course and have a knowledge on this. So this is again the language which we need, Python or R. The Python is more easy to learn when compared to R, which is difficult to learn, but it is very good for statistics, which has large number of libraries which we can work. Similarly, Python also lot of libraries and communities there to support. So with this, this is the resources one can go through. This is a very good review article by Vamadevan et al. On application of machine learning in drug discovery and development. And for to learn about the different artificial intelligent algorithm and their concept, you can refer to this, in particularly the computer assisted drug discovery. And in general, if you want to gain input on artificial intelligence, particularly for the medical professionals, this is very good. And for ADME tox prediction, there is how we can apply to ADME tox prediction. There are a variety of reports available and reviewed by Maltrola. And then we have, at present, we have COVID-19. Lot of artificial intelligence based techniques are being used, and they are being, this is a very recent one where they have summarized all the artificial intelligence based techniques used against COVID-19. So with this, I end my presentation, and I wish you all the listeners a happy Diwali. And also now I hand over the session to Eunice for a question and answer session.
Okay. Thank you, Dr. Murthy. It was really a very informative and interesting presentation. You try to put it together in a very short time. I know it's difficult to do that, but still it was quite interesting. Now I open up the Q&A sessions. We have a few questions here. First one is from NM Kulkarni, Nagraj Kulkarni. His question is, will AI help pharma companies to shift focus from one drug, one target to polypharmacology?
Yeah, this is a very good question, Nagraj. So, because why we went with one drug and one gene is that previously, whatever the we were following experimental approaches, where we can study only one gene at a particular experiment. So we always try a reductionist approach, wherein we always focus on whether the particular which gene it is acting. But now the case is not like that. As we see, we have a polypharmacology, wherein we study a complex network for a particular disease target. So certainly, going hereafter, AI and ML is certainly going to help in analyzing the complex data from biology, from pharmacology, from pathology, that is clinical point, all put together, we will be having uh, complex data which can be analyzed, put to which help us to analyze that what kind of drug and what kind of interaction does it makes, and how we can able to design drug based on this complex regulation. So in this is the place wherein AI and ML is going to play a major role, and certainly the pharma companies will benefit through it, through the approach of polypharmacology, and also using these techniques such that they will come up with a solution like a drug-like compound in a shorter period of time.
Okay. He also has one more question. Can AI and ML alone solve questions in drug discovery, or it has to be used with other approaches like molecular modeling or anything? I mean, just having AI and ML is good enough.
Yeah, the thing is that AI and ML are the techniques, statistical techniques, wherein you want to predict beforehand a particular, to predict whether the problem which I am working with will bear a fruit or not. So that is with AI and ML. So the question is whether this type of technique alone is sufficient. It is not, because you see the disease is complex, and also the protein drug interactions all are very complex. We need lot of human input, like pharmacologist will be playing a major role in generating the data. But in order to have a go green signal, they will be dependent upon AI and ML approaches. It's just a decision making tool which will help the pharmacologist or synthetic chemist, medicinal chemist, or uh, biologist to go about, given the complexity of the data and large complexity of large set of data. On the other hand, the molecular modeling, QSAR, all they can be used in tandem with machine learning algorithm to find a particular solution. So even for example, I showed about structural based drug discovery with ligand based virtual screening and de novo drug design, drug repositioning. All this can be done with the help of computational tools, because the computational tools only will give you the descriptors, or we can able to give or quantify a particular gene data or a drug data or expression data. We can quantify only with the help of cheminformatics, that is molecular modeling, molecular computer aided drug design only will give you those parameters. You can take those parameters from large set of data. Even if they are complex, you can feed into artificial intelligence or machine learning approaches and can get a solution, the answer which you want. For example, if you are given with one database and you are trying to find an association with a particular drug, first you will go for the association of validate the target with the help of AI, to literature mining, uh, and then you arrive at a conclusion, and then you go for a chemical database. You get the chemical compounds. You convert those chemical compound into strings or in the language which the computer can understand, and then you fit into AML technique and say which of the among the reported drug will have reported drug-like compound will be interacting with the target. So this again here, the computational drug discovery and AIML are both, or the in the weighing balance, both are two different pans. So both of them are essential to come with the conclusion of a particular question.
Okay. Next question is from Pranesh Shetty. He's asking how it is useful for small amount of data that we have in drug development. I mean, if you have a small amount of data, how this technology could be used, I mean, can be used effectively, if I'm not wrong. That's the question.
Yeah, because ANML is for both small data as well as big. Because small data means one thing has to be made clear. Because when we are working in drug discovery program, when we are working with a particular target, the chemist will, at least for any target which we are working, the chemist will have at least 500 to 1000 number of compounds. So that cannot be, that may look like a small compound when compared to zinc database which has millions of compound. But this small amount of data of 100 compounds or even 1000 compounds can be analyzed using machine learning, or artificial intelligence can be put to use. The only thing is that whenever we want to arrive at extracting certain features, because if you are given with 100 chemical structure of 100 compounds and you are asked which of the pharmacophore is responsible for activity, which of the pharmacophore is responsible for toxicity, which bond is responsible for toxicity, or which bond is responsible for solubility, then if you can open an Excel sheet, if you are seeing the ordered compounds, you cannot make any conclusions. Even any software will not give you such predictions. Only with the help of machine learning, you can arrive at a conclusion that this pharmacophore will be essential for interacting with the target, that is a protein. This is responsible for toxicity. This is responsible for enhanced plasma life. So these kind of predictions, provided that you have hundreds of data, 100 data points, it is easy to analyze in AI and ML. It not only does for complex and large data, but small data which are complex to be analyzed by humans with the present computers and the computing system, they can be analyzed effectively by using AI and ML.
Okay. So next is my question actually. So
Yes.
how successful the AI has been in identifying the NCs or NBs or anything in India and abroad? I mean, how successful
So,
we are so far?
as such, uh, the as such reported one, there are very few reports. For example, one such example is halicid. You must have heard about, read in the newspaper, which is a new chemical entity which was completely designed and developed using AI and ML approaches, which is projected to be a drug with a novel action. It is an antibiotic which is developed and designed by using AI techniques by MIT people. Apart from this, we don't have any NCEs coming out because of AI and ML. So the main drawback is like that, because everybody has their own drug discovery program, own data which is not shared between each other. So we don't know how to go about a particular compound. So far, the companies or the pharma companies in India abroad are making this for making small decisions. For example, if they have say 1,000 compounds, can we use ML technique to easily come out with say 5 compounds which I can take forward? Like that, they are working on different aspects, not only with the biology, but also predicting ADME, tox. But most of the techniques which I read from this is it need a strong statistics and prediction ability. So generally, what people used to do is that they have a large data, they predict, and they put a for doing AI, we have a positive data and also have a negative data with this. So this is very important. Learning can be done only with positive and negative data and not with either of the data. So then once we predict a model, you feed into the purposefully you feed in the negative compound, and if it predicts that that particular compound is negative, then your model is okay. So using this model, all the pharma companies or all the one, they are now doing this AML for making short-term decisions. Like I want to look into literature. So far, what are the drug targets? What are the disease gene interactions? And so many small decisions are being made. So many tailor-made decisions are being made with the help of AI and ML. So as such, developing a ML completely, NCE completely through AI and ML, it may take some time, but maybe in another 10 years or 15 years. But people have started using these techniques for making certain critical decisions in drug discovery process and clinical trials. So that is for sure they have started, and many companies have put to use. We may not be knowing, but many pharma companies already partnering with AI companies and they start to put to use. And
Okay.
also, so many, not only in pharma company, in biologics, so we have monoclonal antibodies where my colleague Naraj is working. He models, makes mutations in the antibody CDR loop, and then predict this antibody will be more affinity to the target such that the toxicity of the target that antibody can be reduced. Like that, many small decisions, critical decisions in drug discovery and biologics are now being made exclusive by AI and ML. So,
Oh,
but
glad to
the
know
big
that.
picture to emerge, it may take some time until unless a big company, pharma company, openly discloses that I have used particularly the AI mediated technology to come up with the new NCE. Maybe it will happen in near future. Yes.
Yeah, sure. So again, we have Pranesh Shah with his next question. Basic course or tool can preclinical scientist can learn? I mean, basically, he wants to know what are the basic courses and tools that a preclinical scientist can learn and make use of it in wherever he is working.
Okay. So for performing AI and ML, the main thing which is required is first one is the programming languages. The programming languages here we have R language and Python that I showed in the slide. These are very easy because with the help of the kind of mathematical background we have until high school, we can easily learn this programming language first. Once you learn this programming language, many algorithms are available as libraries. Machine language libraries are there. They are created by professional data scientists. So you can take those modules or libraries and you can use it for a prediction. So the advantage is that you having a pharmacy background, knowing the pharmacy data points very deep, with the help of this machine, if you know how to use this program, namely R or Python, to enter your to process your data and feed the data into the tool languaging tool language module, and then predict. So this much if you know, it will be well and good. So, in order to develop skills, mainly the programming skills, and to be very strong in statistics and probability. So, these two are the main thing because statistics and probability already we pharmacologists we have studied and we are having a good knowledge. The only knowledge which we lack is the programming. The programming is not like C++ or Java or very complex coding is not required. You need not to write complex coding. So only thing is that how to take a CSV file, how to process that CSV file, how to clear that CSV file, that you need to know, and then which technique has to be applied to which data, whether we have to do supervised learning or unsupervised learning. For this, you need to have a basic idea about machine learning, like different techniques. I talked about different models like supervised, unsupervised, reinforced, different algorithms, which algorithm to choose for a particular task. These are the thing you need to develop. So once you learn these algorithms, then you are entitled to do AI and ML in a day-to-day routine drug discovery decisions. So for the time, only is you need not to enroll yourself in big AI course, especially meant for people who do professionals. But you can slowly learn these packages. So many YouTubes and other free videos are available in the internet, which you can use and learn the things which we need, namely the basics of machine learning and also the programming language, which you can use for doing your modeling and predictions.
Okay. So next question is from Vijay Habali. So this is about the data integrity. His question is, how do we assure the extracted data from the public domain is valid or authenticated? I mean, basically, he wants to understand if data is not authentic, and if you plug in that kind of data into any model, the outcome obviously would be erroneous. How do you make sure it's not so?
Yeah, that's why that is a good question. Another good question. So we have a data from public databases. So it depends upon how you take the data, how you trim the data. So there are various procedures are there published. You can look for. So whenever you take a chemicals database, say for example, a public domain zinc database, you are taking 1,000 compound which are having a role in which are role in type 2 diabetics. To some of them are good, some of them are bad. So you need to pre-process the data, applying certain uh, modifications, like you have to convert them into a particular string. During that process, like when you do a molecular modeling, you do a energy minimization and other things and all you do. Like that, here we need a data preparation is there, where we need to do, and we have certain metrics for this data preparation. And when we make sure that this data is perfectly okay and fine, then you can use this to train your model. It is not like that you directly take and directly feed into it. So the main thing is that the how you choose the model, how you train it is very important, because the prediction will be made only the type of learning it can make from the data which you feed. So if you don't feed, if you underfeed the data or overfeed the data, then again the problem is the prediction model is going base. So taking all into consideration, you have to pre-process the data using certain metrics which will be helpful for choose making choice that this data is good and I can proceed further with my modeling and training and prediction.
Okay, so next question is from Seram also. Please suggest how a science background person can improve to develop these skills. I think you already answered while
Answered,
answering
yes, yes,
Pramesh Shetty.
yes.
Yeah, the Mallikarjun Jaji wants to know, in India, how many companies or institutions are conducting AI ML training course? I mean, who is providing the course in India?
Yeah, because to start with, even many private universities, they have started with the AML course, because not only useful for drug discovery, for data analytics, they have many universities are started. The one which recently was with IIT Madras, they have three type of courses. One is certificate course, diploma course, and bachelor's course. Depending upon, you need not to, you can join the course and IIT Madras, and if you can have a less money like 40,000 or something, you can finish the certification course in the AI and data analytics. And if you want to still further more interested, you can go for diploma and bachelor's course. Like that, many companies, universities are offering courses. That you can learn. Apart from Indian universities, there are many online platforms are there wherein you can learn each Coursera is one of them. EDX is another one. So in which you need not to enroll yourself in a particular college and go for classrooms. Here, the advantage in online is that you can work at your leisure time. You can learn, and if you miss something, you can rewatch it. So lot of learning can be made from online courses, and they have lot of projects are also there. There are a lot of projects which are available uh, for drug discovery, because you take a data, you model it, and you can yourself train yourself. Training is very much important. You have to keep on trying, training in spite of attending courses. So both online as well as several institutes in India, private universities like VIT and also SRM. There are many institutes. They are also providing this data AI courses. Even Pune University, many are providing these courses which you can enroll. But on the other side, you can also enroll very easily with the Coursera or other thing. And then to start with, what I am doing is that I am started with the NPTEL and doing R programming and biostatistics. And then Python you can learn from web, like online classes. The free courses are also there. There are so many small projects, codes are there which you can able to take and you can do. So it's not like that it is very big and very programming and other things are required to do this analysis. The kind of mathematics and biostatistics knowledge which we have, we can easily do and understand. Once you understand, then it will be very useful for you to make decisions to grow up in your career. So if you go up in your career, when you become a vice president or something like that, then if you know these techniques, you can easily at that time use these techniques to arrive at a conclusion whether to go with this target, this disease, this molecule. So it will be very helpful for you, Malikarjun.
Okay. So yeah, this is the last question. This is from Satyanarayana Chintanipula. Can you explain the scope for serendipitous drug discovery using AI?
No, serendipitous drug discovery. We have certain drugs in the past we have discovered by serendipity, and now they are growing very well. But now we have moved completely into rational drug discovery. So we have a
Yeah.
computational and AI and ML techniques. So these things, serendipity is not. We can't say that it is serendipity, but we can predict unknown, for example, unknown drug, unknown pharmacophore, unknown backbone to be a potent compound against a particular disease. It is not a serendipity, but you are harnessing the computers and ML to identify that particular thing. So in this case, the molecule may be extremely new or novel. It will be. So then you have applied scientific tools to arrive at it, so it cannot be serendipitous. On the other hand, we have lot of what you can say, uh, databases or what I can say. So lot of uh, hints or lot of data from, already we have generated lot of data from variety of experience we have. So the drug which have failed, drug which have performed better in preclinical, which have failed in clinical. So we have lot of experience. We can use this experience properly, document categorically data, uh, put it into a data format. So then the discovery becomes very easy. Even you can have a novel drug. Particularly with the COVID, we have so much drug repositioning has taken place because of AI and ML only. So, for example, even doctors have found out dexamethasone is repurposing dexamethasone for severely ill COVID patient by trial and error method. Some has found like formatidine, like ivermectin, they are found by experience only. But on the other hand, in the near future, we will have lot of, even though AA has predicted several compounds, several companies are pursuing small molecules for treatment of COVID, but even then they are not come up with the preclinical or clinical stage, but several predictions have been made. The compounds are entirely novel. We don't know those compound what action it has. So the repurposing for the drug, it has AI has a large scope in repurposing the drug. So we have around 10 raised per 8 molecules synthesized the whole world and sitting as in our databases. So we can harness this and use this. Even natural products, medicinal chemistry, naturally compounds can be natural bioactive molecules can also be harnessed for things. We can't call it as serendipity, but a scientific way of predicting and making the predictions before doing experiments. That's what I can say.
Okay. Thank you, Dr. Murthy. It was a very wonderful presentation, and I hope the audience have got their starting point if they are interested to further their knowledge into AI and machine learning. Thank you so much on behalf of PERF. We'd like to end the session here. Thank you all participants, and we'd like to know. We'd like to. We will inform you about the future webinars in the WhatsApp group that we have created. If you're part of it, you'll get the links in the future. Thank you, Dr. Murthy.
Thank you very much, Yunus and Nagraj.
Thank you, Murthy. Thank you.
Thank you.